Hello, and welcome to the Chapter 17 lecture on gene expression. You should use the information in this lecture to complete the Chapter 17 guided notes, which of course you should complete before you attend class. In the last chapter, you learned how DNA is put together, how it's stored inside cells, and how cells can make copies of the DNA. But we haven't really discussed the function of DNA yet. So what's it actually for? What does it do inside living cells? Well, DNA represents genetic information, so that means it stores information. What's that information for? Well, the information inside a gene, which is a short section of DNA, can be used to make other molecules, including RNAs and proteins. Both RNAs and proteins have other specific functions inside the cell. So the DNA is like a recipe book for building other functional molecules. The process of using the information in a piece of DNA to make a protein or an RNA product is called gene expression. So when a gene is turned on and is being used, we say that gene is being expressed. Now, proteins and RNAs represent the workforce of the cell. These molecules do all the little jobs that keep the cell alive and allow it to perform its function. Different cells are going to make different proteins at different times, and different types of cells are going to make different proteins than one another. For instance, a red blood cell contains hemoglobin. That's the protein that allows it to carry oxygen in the blood. But a muscle cell doesn't contain hemoglobin, it instead contains actin. That's a contractile protein that allows it to contract. This all relates back to a genetic basis. Because the information for making these molecules is stored in the DNA, by controlling which genes are turned on at which times, the cell can control its workforce and therefore control its functions. Gene expression consists of two processes, transcription and translation. Transcription occurs in the nucleus of eukaryotic cells. During this process, a gene will be read and a copy will be made of it. So that's what transcription literally means, is to make a copy. When I'm talking away up here making this lecture and you're writing some of that information down, you're actually transcribing what I'm saying. So in transcription, we make a copy. That copy is a piece of RNA. That transcript will then leave the nucleus and enter the process of translation. During translation, the piece of RNA will be taken in by a ribosome. The ribosome will read the information and use it to build an amino acid sequence, a protein. Now, during this process, because we're going from the language of nucleotides to the language of proteins, we're actually translating from one language to another. That's why it's referred to as translation. We can summarize this entire process by saying that DNA leads to RNA and RNA leads to protein. Sometimes this rule is referred to as the central dogma of molecular biology, because if you can control this process, you can essentially control the cell. To help you understand this overall process, I have this really silly analogy. Let's say that instead of a protein, that what you actually want to make is this delicious pie. Pies are kind of complicated to make, so you're going to need to approach this in two stages. First, you need to get the recipe. That process is called transcription. And second, you need to build the actual pie. That process is called translation. So first, let's get the recipe. Let's say that to get the recipe, you need to go to the library, which in this case is a nucleus, and you need to check out the book of recipes. The book that contains all of the recipes for all of the pies in the entire world is the DNA. Now that's a gigantic book and you don't want to carry it. So what you're going to do is make a Xerox copy of the specific recipe that you want. Inside the cell, this is going to be represented by a piece of mRNA, messenger RNA. Now that you have your Xerox, you can go outside of the nucleus and start translating this recipe into an actual pie. Now let's say that this recipe is particularly complicated and you need some help. So what you're going to do is find a chef to help you out. The chef represents the ribosome. What the ribosome does is he gathers up the ingredients and puts them together in the right combination to make the pie. That's translation. Now, the chef has a little assistant. It's a little molecule called a tRNA. And what the tRNA actually does is pick up specific ingredients and takes them to the chef in the right order so that the chef can make the pie. So at the end of this, you've got your amino acids put together in the right sequence. That's going to make a protein, or your finished product, the pie. The process of gene expression differs a little bit in prokaryotic versus eukaryotic cells. In prokaryotic cells, the process is much more straightforward and much simpler, and much faster. Why is this? Well, remember that prokaryotic cells lack membrane-bound organelles, so all of the machinery of the cell is kind of thrown together in the cytoplasm. This means that as soon as a gene is transcribed, translation can immediately begin. So these guys tend to be really fast at expressing their genes. It also turns out that the genes of prokaryotic cells are a little bit more organized than those of eukaryotic cells. So again, the cell can access their genes a little faster and complete this process faster than eukaryotic cells. We'll talk about the reasons behind this in Chapter 18. Eukaryotic cells do contain membrane-bound organelles, so this means that transcription and translation occur in different places, and this slows the process down considerably. Transcription occurs on the DNA, so it occurs in the nucleus, and that's kept separate from translation. Once the transcript is made, it has to actually leave the nucleus through a pore, find a ribosome that's either going to be on the ER or floating out in the cytoplasm somewhere, and then translation can begin. So eukaryotic cells are not as fast at expressing their genes as prokaryotic cells. Here's a quick overview of how the genetic code works. We start with a nice piece of double-stranded DNA. Now, only one of the two strands of this DNA is going to hold the actual information, the actual gene. So in this case, let's say it's this bottom strand. What we want to do is make a copy of that bottom strand. We want to Xerox it or transcribe it. So to do that, we have to open up the two strands of DNA, and then we're going to add RNA nucleotides to the DNA so that it's complementary. So here we have a T in the DNA, we're going to match that with an A in the RNA. Here we have an A in the DNA, we're going to match that with a U, a uracil, because this is RNA. Remember, there are no T's in RNA. And so on. Now this little transcript that's been created, this piece of mRNA, can head out to translation. To translate it, what we're going to do is break it down into groups of 3. These groups of 3 are called codons. The cell is able to read each codon, it's like a word in the language, and it knows which amino acid it stands for. So the presence of that codon AUG tells the cell that it needs to put the amino acid methionine at the beginning of its protein. Next, we have this other codon, and that's going to tell the cell that it needs to put this other amino acid in that position. The third codon tells the cell which amino acid to put in the third position, and so on. Now, cells are able to decode the codons all by themselves, but how do we know which amino acids each codon stands for? Well, for that, we're going to use a handy-dandy chart. Let's say, for instance, that we have our first codon, AUG, and we want to know what amino acid that's going to translate into. To use this chart, I'm going to find my first letter A on the left-hand side of the chart. I'm going to look for, here's the A's. Next, I'm going to look for my second letter along the top, U. Here's U. That's going to lead me to this block. And then I can look in the block and find the codon I'm looking for, AUG. AUG codes for the amino acid methionine or MET. And yes, it is just fine to use the 3-letter abbreviations for the amino acids. You don't have to memorize this chart, and you don't have to memorize the names of all the amino acids. Next, we're going to examine the machinery that the cell uses to carry out gene expression. Several of the most important machines are made of RNA molecules. So just to remind you, RNA is single-stranded. That means that it has a single sugar phosphate backbone and that its bases just kind of stick out into space like this. Another important feature of RNA is that it uses the base uracil instead of thymine. So you'll see lots of letter U's in an RNA sequence, but you won't see the letter T. Keep that in mind. Because RNA is single-stranded and its bases just stick out into space, it means that RNA molecules can fold over on themselves and form three-dimensional shapes. Here, for instance, we have a piece of RNA that's folding on itself. And notice that these two little sections are complementary to one another. So they can actually grab one another and make hydrogen bonds, and that helps hold the shape together. Because RNAs can take on a three-dimensional shape, many of these molecules have specific jobs inside the cell in the same way that proteins with specific three-dimensional shapes have specific jobs. Three types of RNA are going to participate in gene expression. The first type is called messenger RNA, or mRNA. It actually carries the message from the DNA out to the process of translation. So this piece of mRNA is the actual Xerox that's made based on the recipe of DNA. Notice that it's just a long strip of RNA with its bases sticking out. It's not folded into a particular shape. Ribosomal RNA, or rRNA, is complexed together with other proteins to make the ribosome. The ribosome consists of two subunits, a large subunit and a small subunit that fit together. The ribosome, if you remember, is the chef in our analogy. So it's actually going to read the mRNA and it's going to use that information to build a protein. We'll see how it does this later. Transfer RNA is also known as tRNA. This is the little assistant from our previous analogy. As the name implies, tRNAs are going to transfer something. What they're going to transfer are specific amino acids over to the chef, to the ribosome, so that the ribosome can put them together in the right order to make the specific protein that we want to build. Now, it just so happens that tRNAs are T-shaped. That's coincidental, but it makes it a lot easier to remember. Transcription is the process of making a copy of a piece of DNA. Our copy machine in this process is this enzyme, RNA polymerase number 2, or RNA poly 2. What this enzyme complex does is it reads the piece of DNA we're interested in, and it uses that information to build the piece of RNA. It actually Xeroxes or copies that gene. Unlike the enzymes we discussed in DNA replication, RNA polymerase II is multifunctional. It performs all the jobs of transcription. It actually opens up the DNA by itself, makes the RNA copy by itself, and then seals the DNA back together at the end. So this process doesn't require helicase to open it. It doesn't require ligase to close it. RNA polymerase II does everything. There are three phases to transcription: initiation, elongation, and termination. Let's take a closer look at each phase. During initiation, the machinery of transcription is going to assemble at the beginning of the gene. It's going to assemble on an area that we call the promoter region. Now, the promoter region is a little section of DNA that doesn't code for anything. It includes a section called the TATA box that's just a series of thymines and adenines that repeat over and over and over again. This area provides a point of attachment for the machinery. So it provides something for the RNA polymerase number 2 to grab onto. Another thing to keep in mind is that even though DNA consists of two complementary strands, only one of those strands will contain the genes, the useful information. DNA polymerase number 2 will attach to both strands, and it will work along both strands, but it's only going to copy the useful strand, and it copies it from the 3' direction to the 5' direction. The strand on the other side is really just there to help hold that piece of DNA together. So it's a structural piece of DNA, but it doesn't contain any actual genes. The giant machine that assembles during initiation is called the transcription initiation complex. This machine is going to assemble on the promoter. Here you can see the promoter with its little Tata box. The first components that assemble are molecules known as transcription factors. They actually help RNA polymerase number 2 grab onto the promoter, and it helps it open up that first little section of DNA. If any of the transcription factors are missing, transcription will not occur. So this entire machine has to assemble before you can enter the next phase of transcription. The second phase of transcription is known as elongation, in which the DNA will be read and the mRNA copy of the gene will be made. To accomplish this, RNA polymerase number 2 zips along the gene from the 3' end towards the 5' end. It opens up the DNA in a little bubble, and then it sucks in these free RNA nucleotides that are floating around, and it matches them with complementary DNA nucleotides. Here you can see a uracil being matched with an adenine. Here's an adenine being matched with a thymine. Here's a cytosine being matched with a guanine, and so on. As the nucleotides are added, the little strip of RNA that's formed will feed out of the back of this enzyme. During the third phase of transcription, which we call termination, RNA polymerase number 2 is going to hit the end of the gene, and at this point all of the machinery is going to fall apart. RNA polymerase will go off to find another gene to transcribe, the DNA will seal back up, and the little piece of completed RNA, which is called a transcript, will now go on to translation. Two different kinds of DNA sequences are used to end transcription. In eukaryotic cells, the machinery of the cell stops transcription once it hits the stop codon. But in prokaryotic cells, a section called a terminator sequence is used that usually occurs just beyond the stop codon. Once transcription is complete, the mRNA transcript can move into the translation phase. In prokaryotic cells, such as bacteria, this process is fairly straightforward. The transcript has been produced directly in the cytoplasm, so all it needs to do is find a ribosome attached to it, and translation can begin. In eukaryotic cells such as our own, however, there's an extra processing step that has to happen to the transcript before it can leave the nucleus. Special molecular caps and tails are going to be added to the ends of the transcript. This is done for three reasons. First, the addition of these molecules helps protect the transcript from enzymes that might otherwise chew them up. Second, these caps and tails help the transcript leave the nucleus through one of those big complicated nuclear pores. And third, these caps and tails help the transcript attach to a ribosome so that translation can begin. Two different kinds of alterations are made. To the 5' end, a molecule known as GTP is added. GTP is a chemical cousin to ATP. It just has a guanine attached instead of an adenine. To the 3' end of the transcript, a long series of adenines will be added. This is known as a poly-A tail. And this should be poly-A tail, not ollie-A tail. The middle of the transcript is also altered through a process known as RNA splicing. In this process, sections of the transcript known as introns, which we also used to call junk DNA, are actually snipped out of the transcript. And the remaining sections, which we call exons, are going to be stitched together into a final transcript that will go on and be translated and used to build a protein. Now, why in the world would you want to chop up your transcript like that? Aren't all of the letters of the sequence important? Well, I want you to think of that transcript as being like a recipe for a set of proteins. You can actually make more than one protein from the letters used in each transcript. This would be sort of like looking at a recipe book and maybe just looking at the chapter on soups. Think about all the ingredients you can put into a generic soup. You could put chicken or beef or shrimp or fish, all kinds of vegetables, different kinds of broths, even weird stuff like beer and wine and noodles. Now, if you put all of those ingredients into one big soup, it's probably going to taste really weird because it's kind of overwhelming. So what you do instead is you look at the recipe and you decide which ingredients are going to stay in, these are the exons, and which ones you're going to leave out this time and maybe use next time. In this way, the cell can alter the transcript. It can alter the recipe a little bit to make a specific kind of protein from that recipe. And the next time it decides to use that gene, it might snip different introns out so that it can alter and make a different protein the next time. This gives the cell a lot of control, and it allows it to make multiple proteins from a single gene recipe. Here's another way to think of it. Let's say you're doing a presentation for class and you want to use in your presentation some diagrams that are in your textbook. Now, you don't want to snip diagrams directly out of your textbook, right? Or you'll mess it up. The same thing goes for DNA. You don't want to chop up the DNA or you'll cause permanent mutations. So what you do instead is you make Xerox copies of the diagrams you want from your textbook. That's like transcribing this gene. Now we can snip this transcript up however we want and we won't damage the original DNA. And so that's what the cell's going to do. Maybe this time the cell decides to keep exons 1, 2, 3, and 5, and it's going to snip out 4. Or it could snip them in a different pattern and make a different transcript. Here it's gotten rid of transcript or exon 3, and it's decided to keep exon number 4. When these two transcripts are translated, they will result in two different proteins with two different functions. So this process gives the cell a lot of control over which proteins are being made at which times for specific purposes. There are several different types of molecular machines that can complete this splicing process, including spliceosomes and some RNA pieces called ribozymes. Spliceosomes are really cool. Let's take a look at them. What the spliceosome will do is it will take that transcript and it bends it so that the intron is bent down in a loop. And then it can snip the introns from the exons at specific points, and then it bends and it puts the two together so that these molecules will join into a single transcript. Here we have the intron section that was cut out. Now that the excess introns have been snipped out and the caps have been put on the transcript, that transcript can travel out to a ribosome and begin translation. Translation includes 3 phases: initiation, elongation, and termination. And yes, these steps do have exactly the same names as the phases of transcription, which is convenient for memorization purposes. However, keep in mind that different stuff is going to happen during these phases, so they're not identical, they just have the same names. Let's take a look at each phase. Ribosomes are the site of translation, so translation cannot occur without a ribosome being present. Ribosomes may be present in the cytoplasm, just floating around free, or they may be found attached to the rough ER. Ribosomes are complexes of RNA and proteins kind of mashed together into this shape. Let's take a look at its structure. Ribosomes consist of two subunits, a large subunit on top and a small subunit on the bottom. The job of the small subunit is to hold a piece of mRNA in place so that the ribosome can read the mRNA. The large subunit has three sort of slots built into it that we call binding sites. These sites are known as the E site, the P site, and the A site. The E site stands for exit, so you'll want to know that term. The P site and the A site also stand for things, but you don't have to worry about their names. Now, what are those slots for? Well, these are sections where tRNAs are going to fit into the ribosome, and they're going to be carrying the amino acid ingredients that we need to make the protein. tRNAs, or transfer RNAs, consist of a long strip of RNA that's been folded up on itself. And there are certain sections that are complementary, so it can hydrogen bond with itself to form this kind of T shape. There are two other regions of the tRNA that are very important: the anticodon on the bottom and an attachment site for an amino acid on the top. Now remember that tRNAs are like the assistant to the chef. They are responsible for bringing specific ingredients over to the ribosome in the right order and at the right time, and they put those specific ingredients in the right place to make the protein that's trying to be made. Now, since we're making protein, the ingredients we're talking about are amino acids. There are many different tRNA molecules, and each one has a specific amino acid attached to its top. So it's responsible for transporting that amino acid and putting those amino acids in the right spot in that protein. Regions of each tRNA, known as an anticodon, help that molecule figure out where it needs to drop off its specific amino acid. Remember that the tRNA is just made of a strip of nucleotides that's been folded into a shape. And it's been folded such that these three little nucleotides on the bottom stick out. That's known as an anticodon. What that anticodon is going to do is it's going to match with a specific codon in the mRNA transcript. So here we have CUC. That would match with the sequence GAG, or GAG. That's kind of fun. So if that mRNA sequence contains the sequence GAG, this tRNA can bind with it, anticodon to codon, and that tells it that it needs to drop off its amino acid in that specific spot. Here we can see another view of this process. This is the mRNA transcript, and you'll notice that the transcript can be broken down into groups of 3 nucleotides that we call codons. Now we're already kind of in the middle of translation in this diagram. So the ribosome has already added a couple of amino acids to the beginning of this protein, and it added those specific amino acids based on these specific codons that were in the sequence. Now this next little tRNA comes along. It's carrying an amino acid on its top, and it happens to have this anticodon CUC. It just happens to find its matching codon in the sequence, GAG, GAG. And so what that tells the tRNA is that it needs to drop off its amino acid and attach it to this growing chain. And so that tells it to leave that amino acid in that specific position within this growing protein. Let's put all these great molecular machines together so we can see translation in action. The first phase of translation is initiation, and as in initiation of transcription, what we're trying to do in this phase is assemble the machinery. The machine we're trying to assemble is called the translation initiation complex, and it's going to involve the ribosome, the mRNA, and the tRNA all fitting together in a certain pattern. First, we have the small subunit of the ribosome, and what it's going to do is latch on to the mRNA transcript. It's going to position that mRNA transcript so that the start codon is directly in the middle. It's going to be in the P site when the large subunit of the ribosome joins up with all of this. Now notice what's attached to that start codon. On top of the start codon, we have the first tRNA. The start codon always has the sequence AUG, so the first tRNA that's going to hook up with it will always have the anticodon UAC. And if you remember your translation chart, AUG always codes for the amino acid MET, or methionine. So this particular tRNA is carrying methionine on top. MET is always the first amino acid that's added to a protein chain. It's not always necessary for the protein's structure and function, so sometimes it's removed later, but we always start building a protein with MET. Once those pieces of machinery have assembled, the large subunit of the ribosome is going to come in, and it's going to slide into place over the rest of this machinery. It'll position itself so that the first tRNA is in the P site, and so that the first amino acid on top is kind of sticking out of this slot on the top. At this point, the translation initiation complex is complete, and we're ready to go into phase 2. The second phase of translation is known as elongation. What's being elongated in this phase is the amino acid chain. So we're building the actual protein at this point. Now this diagram kind of starts in the middle of elongation, not at the beginning, but we can walk through it. We're going to start with the translation initiation complex being all constructed. There's a tRNA in the P site with an amino acid attached to the top. The next thing that's going to happen is that another tRNA, whichever one matches up with the next codon, is going to come along and it's going to fit into the A site. So now we have a tRNA in the P site and the A site, and they both have their specific amino acids sitting on top. Now we have to do a little shuffling around. The tRNA that's been sitting in the P site was hanging on to the amino acid chain until now. But notice what it's done. It's passed that amino acid chain on to the next tRNA that's sitting in the A site by attaching its amino acid chain to the amino acid on top of the tRNA. Now that it no longer has an amino acid on top, that tRNA can leave. So what will happen is that all of these components will scoot over by one codon, by one series of 3 nucleotides in the mRNA. That's going to put the original tRNA in the E site. And remember, E stands for exit. So this little guy is going to leave. He's going to go find another amino acid and attach it to the top so it can do the job again. The tRNA that is now in the P site has the amino acid chain attached to its top. This cycle will now repeat over and over and over again to continue growing that amino acid chain. It'll start with a tRNA in the P site, another one will join it and bring its little amino acid with it. The amino acids will transfer from the first tRNA over to the second tRNA. Here it's attached to the second tRNA. That allows everything to scoot over by one codon so that that original tRNA can leave, leaving the amino acid sequence attached to the tRNA that's now in the P site, and it starts over again. This will continue until the stop codon is reached. Once that stop codon slides into the ribosome, the third phase, known as termination, can begin. Now the stop codon doesn't actually code for an amino acid, so it won't match up with a tRNA carrying an amino acid. Instead, it will combine with this molecule known as a release factor. Release factors actually carry a little molecule of water on top. So what that will do is attach a little molecule of water to the amino acid chain, and that causes all of the machinery to fall apart. So here the machines have fallen completely apart. The ribosome can go off and grab another mRNA to translate. The tRNAs can go get refilled with amino acids and do their jobs. Sometimes this mRNA is reused and retranslated to make more of the same protein, or sometimes it gets broken down and recycled, depending on the needs of the cell. The end of the termination phase marks the end of translation and the end of the entire gene expression process. At this point, several different things can happen to this little amino acid chain that's been created. Sometimes it can fold up into its final shape and go off and just do its job straight away. Other times that amino acid chain represents a small polypeptide, and that will need to be added together with other polypeptide units to build a bigger protein. Sometimes these amino acid sections also need to go to the Golgi apparatus, and as they travel through the Golgi apparatus, they finish folding, and they have other things added to them, such as carbohydrates, that are necessary for doing their jobs. At any rate, that protein has been created, so it can go off and do what it needs to do. All right, you guys, you made it. You made it all the way through the process of gene expression from DNA to finished protein. Make sure you fill out your notes thoroughly and bring them to class. Jot down any questions you have, and we'll work with these molecules in class together.